just a little bit about our research here at UQ in my lab. And this is around looking at lactic acid bacteria, developing or discovering new lactic acid bacteria starter cultures for plant-based dairy fermentations. And this is a collaboration between the University of Queensland and Denmark Technical University. So myself, this is Wen Kang is the PhD student, and Sangeeta is a food sensory senior lecturer at UQ. And we had an agreement with Denmark or DTU that we would have one PhD student at UQ and one PhD student at Denmark. And this is Anders, who's in Denmark. And Klaus is the supervisor over there. And the idea is that we can exchange ideas and the students can go to each other's labs and so on. So Wen Kang is going to Denmark next year to do some of his project there. And we have regular meetings with the Denmark team. But it's a nice thing where we're both looking at this, characterizing or discovering and characterizing new bacteria to ferment plant-based foods. So what are plant-based foods? Well, this is what they used to look like 20 years ago. They were plants. And you go to the supermarket now, and this is what plant-based foods look like now. So they look like meat. They look like meat sausages, meat cheese. They look like egg. All this sort of stuff, yogurts, milks. So there's a huge interest in plant-based foods these days, and a growing interest, because of greater health awareness and also concerns about animal welfare. There's a lot more people, are not strictly eating meat, not vegetarians, but more flexitarians, where they're kind of moving to a better mixture of plants and animals in their diet. People have concerns about environmental impacts of animal production, greenhouse gas emissions, particularly for cattle, for red meat. Dairy industries also, the dairy foods have also had issues with people have allergies to milk proteins, and a lot of people are lactose intolerant as well. So they're looking for alternative foods. So there's a lot of plant-based milk alternatives out there. I'll call these PBMAs. And when we call them milk, they're not really milk. They're not from an animal. So we shouldn't really call them that. But the market of PBMAs is about 27 billion around the world. So soy milk is probably the main one. Soy milk is the main plant-based milk alternative out there. And these milks constitute about 15% of the total milk market. So the 15% of compared to say cow milk. So these dairy-free milks. So in Australia now you can get milk from almost anything they can get milk out of. So soy milk is the main one. Almond milk is getting big, but there's also a bunch of others: oat, rice, coconut, cashew, macadamia, walnuts, and so on. So we were interested in almond milk initially for some of our work. We've kind of moved into soy a little bit more, but the market share in Australia for almond milk from 2015 to 2020, soy milk has actually fallen in popularity. Soy has a taste that Western palates are not really used to, and it can be objectionable from some. The amount of soy milk is reduced from 69% to 48% of the PBMA market share, whereas almond milk has increased from about 18% up to 44%. So it's almost the same as soy milk now. So they're selling almost the same amount of these two milks in Australia. And a lot of this goes towards coffees. So people who drink coffees, their cappuccino, they have almond milk in there. But you guys are nutritionists, and you'll know that almond, when you look at the nutritional content of this, it's much worse than soy milk. The protein level is much lower, and also much worse than cow milk as well. So even though the cost is similar and the production is similar, or the consumption is similar, the nutritional density of almond milk is quite watery, very low protein, very low sugar or fat, and so on. But it's healthier, but it's just because it actually, there's only about 3% of almonds actually in this milk. In the production stakes, almonds are actually a large industry in Australia. We actually produce quite a lot of almonds. We grow them in the almond trees. We're the second largest producer in the world. The US is the first. They produce about 90% in California, and we're second, but we only produce 8%. So it's quite small, but in the scale of things, it's reasonably large. About $1.6 billion worth of almonds are produced in Australia, grown as a crop, and we export around $550 million worth. So it's Australia's most valuable horticultural export. Almond milk as well also has a nutty flavor, and it's not it's different to milk, and people perhaps are not as interested in almond milk just because of the flavor. So we were interested in, can we use fermentation to enhance the flavor and texture of some of these plant-based milk alternatives? So almond milk was one example. So can we use fermentation to make new sort of yogurts or cheeses based on some of these plant-based products that have improved flavor, texture, and so on? And I showed you this periodic table before, so we don't need to go through this, but the point I'll make is that which fermentation organism are we going to choose? And the green ones are the most common, which is the lactic acid bacteria. So that was our focus with this, but we're also doing a little bit of work with some of the molds as well. So I guess pairing some of these new bacteria or microbe and food combinations and making new foods. So we're interested in lactic acid bacteria. It's what I did my PhD on, how long ago? 25 years ago. And these are non-pathogenic, generally regarded as safe organisms. And they use a lot in food fermentations, as we saw on the table. There's some strains of probiotics, but they're also naturally present on fresh foods. So if you just take any fruit, vegetable, or meat product, and you try to isolate lactic acid bacteria, even if they're not fermented, you'll find them there in low numbers, hundreds to thousands. So you find them in high numbers in fermented food, so kimchi, yogurt, and so on. But also just fresh foods like this, you'll also find these bacteria are present on the surface of these foods growing in the fields. Some produce antimicrobial compounds. They produce organic acids like lactic acid, but other things like small peptides or hydrogen peroxide. And they also produce flavor and texture compounds as well. So we're interested in using some of these plant-based lactic acid bacteria to see whether they could ferment these plant-based foods. But we also were interested in just using some of the dairy, the cheese-making Lactococcus bacteria. So can we use these, say, just strains that are already used for cheese making? Can we use them in sort of almond milk to ferment the almond milk? So let's test that first. So we put these bacteria, the cheese bacteria that ferment normal milk. So where is it? Strain A in cow milk. So we put the bacteria in normal milk. The bacteria will produce acid, so you get pH reduction. The cow milk's around 6.5, and you get acid being produced very quickly, like you would with yogurt or cheese making, down to around 4. If you put this same strain in almond milk, in the blue line here, the pH doesn't change. It stays pretty much the same, up around 6, above 6 here. So it doesn't produce any acid. So the typical sort of dairy strains that are used to make cheese and yogurt are not very well suited to these plant-based milks. There's a question here, do fermented soy milk and almond milk have a longer shelf life than regular milk? Yeah, they would. Yep, that's right. Because the pH is reduced, there's been acid being produced, and so on. Yep. It has more preservation effects. And when we also tried some yogurt starter cultures as Streptococcus, and it produced acid a little bit, but it wasn't that good either. So what we're interested in, so with these plant-based yogurts that are on the market, so there are some out there, they typically add in sugar in there. They'll add in glucose to allow these bacteria, these dairy cultures to grow and produce acid. But we wanted to find some lactic acid bacteria that could ferment these plant-based dairy alternatives. So we asked the question, can plant-based lactic acid bacteria better at acidifying plant-based dairy alternatives than some of these dairy bacteria, because the plant-based lactic acid bacteria are naturally present on plants on the surfaces. So presumably they can actually, feed off some of the plant material and actually grow in that environment. So we had student projects over the last, about 10 years ago, we had students that isolated bacteria, lactic acid bacteria from fresh fruits and vegetables. We had a collection of around 800. Lots of student projects going to the market, going to the shops, bringing in fresh foods, and we would then take samples and then plate it out onto some agar and then isolate bacteria. And then more recently, we then got those, our culture collection, which is around sort of 600 now. And we then put them into almond milk and then see whether they can produce acid in the almond milk. So can they grow in this almond milk? So we screened around 600 plant-derived lactic acid bacteria, and we put this pH indicator dye in there as well. So we added this color indicator. So almond milk looks like this. When we add the pH dye, it looks purple. And if there's acid being produced, it will turn a yellow color, indicating the pH has fallen down. We also measured the pH after 24 hours. So we took 3 mils of these unsweetened almond milk. This is just pure almonds in water. This is UHT, so it's sterile, and we put them 3 mils into these little tubes, and we did this 600 times. And we put out a single pure culture of bacteria into each, and then we looked at the color change, and we also measured the pH for 223 as well. And you see that some strains are unable to produce acid, whereas other strains are able to produce lots of acid below pH 4. And when we look at the acidification over time, we find that some strains are very strong acid producers. Within a few hours, the pH can drop very quickly down to 4. Some are medium acid producers, and some are unable to produce much acid at all. So we were interested in understanding, what are these ones here? What are these species? What are these ones here? And why these ones are able to produce acid and these ones are unable to do that? So we're obviously looking at the sugar composition of the almond milk, and it's very different to cow milk. Cow milk is mostly lactose, whereas almond milk sugar composition is mostly sucrose, but it also has some glucose and fructose. We did some sugar analysis, and it's mainly sucrose. And we looked at the pathway for sucrose metabolism. So in the bacteria, sucrose is a disaccharide, gets transported in, there's a transporter, SACB, gets phosphorylated, and then it gets cleaved into the monomers, the single sugars, fructose and glucose, and this fructose gets phosphorylated, and then this enters into the glycolysis pathway to lactic acid here to produce energy. So we did some genome sequencing of our bacteria, and we were focused on the ones that produced acid. And we found they had this a gene cluster in there that had the genes that produce this, the SACB, SACA, and SACK genes. So SACA, B, and K, and also a gene regulator here. So these 4 genes were found in the ones that could ferment the almond milk, but we also checked another one. These are Lactococcus strains. One that was unable to ferment the almond milk didn't have these genes in the genome. And then we were also interested, so we sort of found a correlation between sucrose genes and the ability to ferment or produce acid in almond milk. But then there's a correlation there, but we really wanted to show for sure that sucrose genes are essential for almond milk acidification, and also other plant-based milks as well, which have sucrose in there. So we wanted to make a spontaneous sucrose negative mutant. And we tried to do that using genetic manipulation techniques, basically to inactivate one of these genes. But my student, Wang Kang, spent 3 months on that and we couldn't get it to work. So we thought we'll just do something simple. We'll just pick colonies and plate the bacteria out onto agar and have sucrose in that agar and have a pH indicator in there as well. So when you streak out, we have the color indicator that we had in the milk. So it's purple. So the agar looks purple. But the bacteria, when you streak it out, it looks yellow, which means it's producing acid, which means it's using the sucrose that is in that agar. So this agar has high levels of sucrose, has a pH indicator. The colonies typically will look yellow. He then plated out 50,000 colonies on agar, lots of plates, about 250 plates, and then he got I think he got 3 or 5 mutants. And one of the mutants look like this. So if you streak it out on the agar, you see that it's not yellow. It can grow. There's a little bit of glucose in the media, so it can still grow, but not enough. It doesn't have enough, doesn't produce enough acid to make the colonies look yellow. So we're able to do a screen of 50,000 colonies and try to find ones that had lost the yellow color. And this is one of them showing here. We got 5 of these. We did genome sequencing on these ones. And as expected, what we saw was mutations in the genes of the sucrose metabolism genes, deletion events. And we had 5 mutants, and 3 of them had the same mutation here, and we had 2 other ones here. So these will disrupt those genes and prevent the transport. It's a transporter mutation, or the hydrolase mutation here, making these mutants here unable to utilize that sucrose. The rest of the genome is unchanged. They only had 1 mutation in the genome. So it's a very nice, simple system where we can ask the question, is sucrose important for almond milk acidification? So we took the SACB mutant, which was mutated here, 1 base pair deletion, so it's no longer producing the transporter. It can't transport in sucrose. So we wanted to test, can this mutant, is it defective in fermenting some of these plant-based milks? And if it is, then we know for sure that yes, definitely sucrose metabolism is critical. So we took some almond milk, also cashew nut milk, macadamia nut milk. These are high levels of sucrose. We also tested it in rice milk and oat, which have 0 sucrose. It's mainly glucose. And also in cow milk, which is 0 sucrose, because it's mainly lactose. So this is a little bit confusing, but we have our milks here, the almond, cashew, and macadamia. These are high in sucrose. And we have our wild type bacteria and the SACB mutant and no lactic acid bacteria. So this is the pH after 24 hours. So the pH is around neutral when we don't add bacteria. When we add the wild type in the red dots here, a lot of acid is produced here, as we know, because we screened it in almond milk. So this strain is very strong, but the SACB mutant in the in the blue arrows, blue triangles, is defective. It's unable to produce a lot of acid in these high sucrose milks. Macadamia nut has a fair bit of glucose in there as well. It's not all sucrose, so it's able to use some of that sugar to produce some acid. But in the cashew nut milk, it's 100% sucrose, so that the mutant is completely unable to produce any acid at all. Whereas when we look at rice and oat milk, which has no sucrose, if you knock out the sucrose transporter in that mutant, it doesn't affect its ability to produce acid at all, because it's mainly glucose that it's using, and sucrose it doesn't really care about. In cow milk, it's mainly lactose. This strain is really bad. It doesn't have very good lactose metabolism genes, so it even is unable to ferment that lactose, the normal milk. And the mutants is about the same as well. So obviously knocking out sucrose transporter is not going to affect it. So this is a nice evidence to sort of show that for a strain to ferment and produce acid in a plant-based milk that has sucrose as the main sugar, it's critical to have those sucrose genes present. Otherwise you won't get any fermentation. And soy milk also is sucrose as the main sugar in soy milk, and we also get the same results with soy milk as well. So these genes are really essential. If you want to find a culture that can ferment some of these plant-based substrates, you've really got to have sucrose genes in there. Just quickly, the other thing we found when we did genome sequencing, we found the sucrose genes were all here. Some of the strains will look like this. They had the 4 sucrose genes, and they had these nisin genes upstream. This is the nisin gene operon in blue, And this is an antimicrobial peptide that's produced by some bacteria. And it's a food preservative that's also added into some foods. We found some strains, the sucrose genes were close to the niacin genes in some strains here, some of the lactococcus, but in other strains we just found the sucrose genes, but it was missing the niacin genes here. So you see that all these blue gene cluster here is missing from these strains here. These are able to ferment sucrose, but they don't produce niacin. Niacin is an antimicrobial, and when we look at the when we spot the bacteria on an agar plate here, these ones up the top here are these strains up here. There's other ones in the database that we used as well, but these ones, these first 4 or 5 here are our strains. These all have the niacin genes, and they produce a zone of clearing, an antimicrobial zone. We have another indicator bacteria in the background in the agar. So these are all producing the antimicrobial, they're secreting it out, and it's inhibiting the other bacteria from growing. Whereas all these ones down the bottom here are these strains down here, and these are missing. They're missing the niacin genes. They don't have the niacin genes, so they don't produce the antimicrobial chemical around. So we have starter cultures that we can choose from that can ferment these plant-based milks that can produce niacin. So we could choose one of these. It will ferment and produce acid. It will also produce niacin in the milk. Or if we don't want niacin being produced, because if we want to add another culture maybe to produce flavor, we may choose one of these other strains that can still produce acid in the milk, but won't produce niacin, which might kill another culture that we try to add. So this is useful if we want to improve the safety of the food that we're fermenting as well. So this is an antimicrobial substance that will inhibit things like listeria from growing. Just briefly before I finish is talk about there's other milk alternatives, and I don't know whether you've heard about it. It's all the craze in Australia at the moment with precision fermentation, it's called. This is where they're using microbes, genetically modified microbes, to produce proteins present in milk or eggs or anything. Animal-based proteins, casein and whey being produced, and there's lots of companies in Australia and the USA that are developing these systems called precision fermentation. It's kind of using genetically modified yeasts in many cases, or fungi. Australia has one here, Eden Brew, which is expanding, and in the US they have a large company here that's producing products now. And this is the vegan cowboys, where they talk about how they used to use a cow to make milk and then get the casein and make cheese. And now they're using bioreactors or a stainless steel cow to use a yeast to produce the casein protein to make cheese that way. So you can kind of use a non-animal system to produce protein. The problem is the level of protein that you need to make cheese, 25% protein, very difficult to produce using a microbe. They just don't produce enough. But there is positives with this approach, using less water, less land than animals, less energy, less feedstock. And Perfect Day in the US are releasing products now. There's ice cream, Brave Robot ice cream, which has some of this fermented protein that's being produced, the casein that's being incorporated in here. Not very high levels though, quite low, at this stage, and it's quite expensive to still produce it like this. But anyway, this is an area that's growing in expansion, in funding, and startup companies and so on around the world. All right, so conclusion, plant-derived LABs can efficiently acidify almond and other nut milks and soy milk, and they really need the genes to utilize sucrose, and probably because they've adapted to plants, they're not adapted to dairy milk. Most of the dairy-based starter cultures don't possess these genes, so they're adapted to using lactose. And most of the plant lactic acid bacteria are poor acidifiers of cow milk, as we saw in there. They don't have the lactose genes. So different bacteria will have different genes that will allow them to ferment different substrates, and we really need to select and identify the best ones that we can use, not trying to force a dairy strain to ferment plant-based substrates. So we're doing further work. Wen Kang's working on things like texture getting the bacteria to produce polysaccharides, thickening agents, and so on. We've gone back and screened our collection for exopolysaccharide production, and we've got isolates that produce slime, exopolysaccharide slime that can thicken some of the foods, these plant-based foods. And we're also looking at flavor. So we've screened the culture collection for flavor, a key flavor compound as well, and to put these things in with the fermentation to get new flavors as well, some of these plant-based milk alternatives. So acknowledgements, yep. So this is the team at UQ and also the team at Denmark, and it's continuing on further research, and happy to talk more about this if anyone wants to as well. Cool. Okay. That wraps up everything I've got prepared for today. I'm happy to answer some more questions if anyone's got some. Feel free to put them in the chat or unmute your microphone.
Okay, for everyone who still wants to ask question for Professor, maybe you can drop your question on the chat box or kindly raise your hand and then turn on your microphone. Okay, but I think that we already have a lot of questions before.
Yeah,
right?
That's fine. they've been really good questions. Thank you. I've enjoyed answering them. Yeah, they've been tough. I like it though. You're very smart group of students. So well done.
Thank you so much, Professor. Even when I haven't opened up the question and answer session, they already have an active participation to ask questions. Okay, so good. I think there's no question because they already asked many things before. So everyone, please give applause to our Professor Mark Turner for this insightful material on this topic, or you can give your reaction for our professor today. So Professor, many people out there which haven't dived enough into the biotechnology think that when they heard the word microbes, they will think about only the negative, but it actually has a lot of benefit for food industry and for our health, of course. But we also have to pay attention to all the treatment we give to the food so that the fermentation can be done successfully, because it has factors influencing the environment which affect the result of the product. And also about the periodic table, Professor, I personally never know about it before. So thank you so much for bringing up this topic, which opened our mind wider about the fermentation. Very interesting. Okay, so once again,
you're very welcome.
Thank you. Once again, give applause for our professor who has given us many valuable things related to the food fermentation. Based on what Professor has said, I can point out that fermentation is a microbial anaerobic catabolism of organic compounds, generally carbohydrates, in the absence of an external electrode acceptor, involved exposing the raw or starting food material to conditions that favor growth and metabolism of specific and desirable microbes. And fermentation has advantages such as more self-stable compared to the parent food, have unique aroma and flavor characteristics compared to parent food, and also increase the digestibility. And then the microbes used are mostly lactic acid bacteria like Lactococcus, Lactobacillus, Streptococcus, and also propionic acid bacteria, acetic acid bacteria, and also fungi like yeast, Saccharomyces, and then molds, Aspergillus, and Penicillium. And the source of microbes itself can be found from natural fermentation, and also backsloping, and also controlled fermentation. And the fermentation also have factors influencing from the intrinsic, extrinsic, and also implicit. From the intrinsic itself, there are activity, water, pH, nutrients, and also inhibitory substances. And for the extrinsic one, there are temperature and also availability of oxygen. And for the implicit, there are bacterial interaction and bacterial effects. So that was a little summary from our discussion today. Once again, massive thank you to you, Professor Mark Turner, for your willingness to give your time for us. And also, I'd like to say thank you so much for all the participants for your very active participation.
Yes.
As Master of Ceremony and Moderator, I want to say to apologize for any mistakes that I made. But before we close the event today, we're going to take some documentation, Professor. So for those participants who haven't turned